DNA polymerase I cuts out the RNA primer and uses 5' to 3' polymerase activity to replace the RNA nucleotides with DNA nucleotides. DNA ligase seals the nick between the DNA added by DNA polymerase III and that added by DNA polymerase I without adding another nucleotide to the strand. Through these steps, the replication machinery of the cell uses the parental DNA as a template to synthesize the leading and lagging strands.
What is this one?
DNA polymerases catalyze the synthesis of new DNA strands from a DNA template. DNA polymerases require a pre-existing RNA or DNA strand, the primer, to initiate new DNA synthesis. These polymerases add deoxyribonucleotides only to the 3' end of a growing strand. Taking a closer look at the nucleotide polymerization reaction, the 3' end of the primer contains a free 3' hydroxyl group. The 3' hydroxyl reacts with the 5' end of the next free nucleotide to be added. Overall, the new DNA strand grows in the 5' to 3' direction.
Okay, clear up to this. So your homework will be on this one. So mainly the initiation of DNA replication. So all the enzymes, all the proteins, you need to give the functions of that. If you know the structure, you need to show the structures and the replication fork. Okay, that is the main thing. You need to know this one very well because everything is based on this. Now we know this part and the replisome, zydase, the function of those thing, elongation and termination of bacterial DNA with cluster complex, termination locations. There are some kind of termination points, and so this is all about bacterial DNA replication. Now we need to think about eukaryotic DNA replication. It is more or less similar. Eukaryotic DNA replication and bacterial DNA replication is more or less similar, but there are certain kind of things because eukaryotic DNA or eukaryotic cells contains more number of proteins. It is more complicated. It is very long, and that's why they have certain kind of things. So it needs a very short time to replicate the whole DNA. That's why the origins are a lot, not a single origin. There are a lot of origins. And then, as prokaryotic circular, so starting from one point, they are going to the end, that's fine. But in case of eukaryotic DNA, two ends, there are some kind of problem there. Okay. And another main part is that prokaryotic DNA always there is no such histone protein. But eukaryotic DNA, their model is nucleosome model. So there is histone protein and DNA is coiled onto the histone proteins. So the new DNA is formed must have the histone proteins. Okay. So these are three main parts: circular, linear, and it is long, and prokaryotic DNA is short. So multiple origins, endpoint replication, and the histone proteins. Okay. Now it has a lot of origins, and generally yeast is eukaryote, but yeast chromosome is not linear; it is sometimes circular. So they have ARS, that is autonomously replicating sequences there, just like Odyssey in the prokaryotic. In the prokaryote, they have Odyssey region, but in the case of yeast, they have ARS region. That is the origin. And this is just 100 to 200 BP long, mostly AT rich, and another protein is required to bind with this one. In case of prokaryote, we know that DNA A protein that was binding to the ODC region to initiate the synthesis or replication. Here another protein is required that is called the origin recognition complex. So ODC, this WRC protein, so WRC needs to bind with that region, that is the ARS region, then the initiation can start. In case of prokaryote, we had, that is the protein was DNA, protein is small. If you write protein, then protein should be like this, small n, small a. Okay? DNA A and that was ODC. It is more or less similar structure because they have the circular DNA, but yeast is eukaryote and that one is prokaryote, but the structure is more or less same. And the thing is that they have several origin. In case of linear DNA, just like us, we have several origins of replication. From each origin, replication starts and goes to both direction. Okay. Finally, these two replication forks, they can meet together, okay, and creating two separate chromatids. So this is the main, in a nutshell, this is the process of replication. Now, if this is the thing, so we have multiple origins. How many? How many origins are there? The entire genome contains almost like 1,000 of origins in case of our human chromosome. Now the question is that in a chromosome or in a DNA, there are several origins. Yeah. Okay. If everything is activated, these origins, then the replication starts and goes to both directions. That's fine. That was the first thing. So each and every origin needs to be started from here. The replication starts from here, goes to both direction, and they will meet each other, and that will be fine. When this new DNA strand is produced, at that time, suppose another replication is going on, because there are two kind of parts. One is heterochromatinized region, one is euchromatinized region. In case of euchromatin part, it is lessly coiled, so the replication can take place very easily. But in the heterochromatinized part, it is highly coiled, so it would take more time to replicate that part. When the heterochromatinized part is replicating, so if the another cycle can start here, what would happen? Suppose that is the less heterochromatinized part is happening around less synthesis phase, late S phase, but eukaryotic part is earlier phase. When this part is replicating, still replicating, and this is done already, those proteins, if they can start another replication here, then there will be some problem, right? That is one thing. Another thing is that how many origins should start replication? All, or sometimes it is something like that. That is a reserve player in a team. There are, suppose 11 player will play, but we have some kind of reserve player in soccer. 16 other players are there, they are the reserve players. Here there are some kind of things that there are a lot of original sites. Sometimes they are very close to each other. If this one is activated, this need not to be activated. This one. Okay. And there are certain kind of proteins, they can do these things. This is called licensing. Replication licensing factors. So some proteins are there, they will bind to this site, these origins. When they are bound to this site, those origins are activated. Otherwise, they are deactivated. Okay? These are called replication licensing factor. when a site or origin is licensed, then replication will be done there, otherwise not. So what are those things? And when is it done? During G1 phase. So we talked about the cell cycle. So cell cycle, in the first part of the cell cycle is that after division, if it is the divisional phase, then the first part is G1. Okay. Our neuronal cells not divide throughout our lifetime. Okay. So why? Because they don't have that factors for the division. They don't have that signal. Here is one checkpoint. This is called cell cycle checkpoint. In this checkpoint, the cell will decide whether it will go for the division or not. Our neuronal cells are here in this checkpoint. They are stuck here. They cannot go to the other divisions. This is called Z0 point. Okay. And our skin cells are in G0 all the times. Right now, if we have a cut, then it will go from G0 to G1 phase and then it can divide like this. Okay. So in such case, if they will pass the G0 phase to G1 phase, that means they will go for the synthesis. That is the checkpoint. If they will pass that checkpoint, they will go for the division. Division means the first step of division is the DNA replication. DNA needs to be replicated into 2. Chromosome needs to be replicated into 2. Okay, that is the first part. So all this origin recognition complex and licensing of the origin is done in G1 phase. Pass the G1 checkpoint, then the licensing starts. Okay, these are the part of the DNA which needs to be licensed. These origins need to be licensed, and the replication starts from those origins. And they have some kind of things, origin recognition complex, that is the protein, this protein, with other 2 factors. So there are 2 other factors associated with this protein, these 3 as a whole called MCM2 or MCM27 complex. What is this MCM27 complex? It is mini chromosome maintenance complex. That is actually the licensing factor for each and every origin site. Now, the question is that was the problem. So when the replication starts, there is no function for MCM. Okay? If they will move to another part, they can start the replication there. If they can move to the another chromosome, they can start the replication there. But that can be prevented. There are several other proteins are there. When the replication starts, they prevent the MCM27 binding to the DNA. So they will be inactivated. They will not bind to the origin. They will not bind to the DNA. So there will be no other replication until that replication is finished. Okay. Now, in case of prokaryotic DNA, we knew that there are 5 DNA polymerases, and in case of eukaryotic DNA, there are a lot. Of them, there are main 3 DNA polymerases. DNA polymerase alpha. Here we will name them as the Greek letter alpha, beta, gamma, something like that. Alpha, DNA polymerase delta, and DNA polymerase epsilon. Okay. DNA polymerase alpha is like DNA polymerase 1. Has that exonuclease endonuclease activity and can synthesize a short primer, a short stretch of DNA, not primer, short stretch of DNA. And DNA polymerase delta, it is working on the lagging strand. It can synthesize a short stretch even better than DNA polymerase alpha, but that's why it is working on the lagging strand. DNA polymerase epsilon is the main DNA for leading strand. It can synthesize the whole DNA as a whole. Okay. These are the list of all DNA polymerases here. Mainly alpha, delta, and epsilon. They are the main DNA polymerase for the whole DNA replication. These are these gamma, zeta, eta, theta, iota, kappa, lambda, mu, sigma, phi. These are some damage. If there is some mismatch repair, that part needs to be cut out and that parts need to be repaired. In such case, those DNA polymerases can work. Or DNA repair, replication and repair of mitochondrial DNA. So there are so many functions there, but they are not mainly for the nuclear DNA replication. Now the problem with histone. So there are one main difference, that is the DNA polymerases, different types of DNA polymerases. In case of prokaryotic DNA, we found that DNA polymerase 3 work in two strands. Here there are two DNA polymerases, polymerase delta on the lagging strand and epsilon on the leading strand. And the DNA polymerase 1 and DNA polymerase that is alpha, they have the same kind of mechanism. In both cases, that's proofreading and the cutting the RNA primer, stretching that RNA primer, repairing that part. And DNA ligase is doing the same thing, that's the nick join. All these things are more or less simple. Okay. But the main problem here is that DNA is coiled into nucleosome. So it is like that. When a new DNA is formed, here, the nucleosome, that is a histone protein, needs to be replicated. Okay, new histone protein is required there. It should be replicated there. And just after the creation of the DNA, all the times, when a DNA replication is done, the histone proteins needs to be replicated, then can go for the another one. This way. Okay. So here the process is that they can redistribute, they can disrupt that old histone protein and they can redistribute that old histone protein into new one. So there is synthesis of new histone protein as well as the redistribution of the older one. How? This is called the histone octamer and mainly done by a protein that is called the histone chaperones. These are the proteins and they can do the formation of histone proteins and the addition of that histone protein with the DNA, with the coiling and everything, that is the formation of nucleosome. Okay. Now, how is the process? So we know it is H2A, H2B, H2B, H2A, then H3, H3, H4, H4, like this. Okay. Now, when they are breaking these histone proteins, they are creating two different things. Two dimers of H2A and H2B. So one dimer, it is H2A and H2B. Another is H2A, H2B. There are two dimers and one tetramer containing H3 and H4. So this one is breaking into these three parts. Okay. Now what is happening here? This one can go to DNA. They are replicated here by the histone chaperones. So from one tetramer, another tetramer is formed. From one dimer, another dimer is formed. Now the joining is that it is not always true that old one is joined with old one. Old one can join with the new one can join with the old one. In such a way, they are dissociating first into these two kinds of dimers and one tetramer. Then they are replicated into two, and then they are rejoining in this fashion, old to new or new to old. And this assembly of all this new nucleosome is done by CAF protein. CAF is chromatin assembly factor, CAF1. CAF1 is responsible for joining the nucleosomes. Now in case of termination, termination is very simple. Just two replication forks are moving to each other and they just join, fuse with each other. That's the thing. But we really don't know at that time how the helicases are functioning or something like that. We just know that they are fusing with each other and forming the whole thing. Okay, this is a simple termination for particular origins, each and every origin. Now the problem is at the end. That is the telomeric region. At the end of the chromosome or at the end of the DNA. And what is the problem here? That when they are synthesizing those things, like so leading strand is okay, that's fine. New DNA is formed here, so there is no problem. Leading strand is fine. In case of lagging strand, so there is one RNA primer, then DNA, then RNA primer, then DNA. So inside to here first, from here they need to go here, then they can synthesize this part, then the DNA synthesis can be done. Okay. So how this one can be healed, this part. This can be healed in this way. So this RNA primer is removed by DNA polymerase 1 or DNA polymerase alpha. Then from here, there is one open OH group here because open OH group is required all the time. Here is that, then they can synthesize up to this. Okay, there is no problem. They can synthesize up to this one. What happens if this one is removed? There is no DNA here. If there is no DNA here, they cannot synthesize this part because DNA polymerase always need an open OH group all the time. They cannot synthesize this part. So in such case, there is always overhang. One DNA, the new DNA is short and the old DNA is long. And that is called the overhang. All the times there is a 5' overhang here. Okay. Now in this case, what will we have? There is one enzyme, DNA polymerase, any of the DNA polymerase cannot work in that case. There is one special enzyme called telomerase. Telomerase is working there. What is the function of telomerase and what is the structure of telomerase? It contains its own RNA primer. It doesn't need any kind of RNA primer. It has its own RNA primer by itself. Okay. So what it can do that, it can attach to the end of this overhang and just repeat. This is a very much repetitive sequence and extend one repeat. Extend one repeat into this part. It can synthesize the rest of this. We don't know why it is extending that part, but each and every time it is extending couple of repeats there. And then synthesizing all the things. Finally, still there will be some kind of overhang, but that will not hampering too much. So their function is thus, function of telomerase is that adding repeats at the telomeric end, then synthesizing the original DNA strand up to that one. If there are repeats, if one repeat is not there, that will not create any kind of that kind of problem. That's why it is creating that much repeats there. Okay. Let's have a look.
Telomeres are present at the ends of eukaryotic chromosomes. They serve to stabilize a chromosome's ends and provide a solution to the so-called end replication problem of linear chromosomes. Here the chromosome is being replicated. It has multiple origins of replication. Each RNA primer has provided a hydroxyl group at its 3' end for the replication of the new DNA. The RNA primers will be removed and replaced with DNA. The 3' ends of the DNA provide starting points for removing the adjacent RNA and filling in with dNTPs. In some single-celled eukaryotes, the terminal primer is placed at the end of the chromosome, but in humans, it's placed 70 to 100 nucleotides from the end, so that when this primer is removed, an even larger gap remains unreplicated at the end. The space left at the end of the chromosome cannot be filled in because at this location, there's no free 3' hydroxyl group to provide a starting point for DNA polymerase to add dNTPs. A gap remains here, as well as at the other end of the linear chromosome. Without a special mechanism, DNA replication always leaves a gap at each end of linear chromosome, shortening the telomeres with each replication. This is the end replication problem. In some cells, the single-stranded protruding end of a telomere, known as the G-rich 3' overhang, can be extended by an enzyme called telomerase. Telomerase possesses protein subunits as well as an RNA component. The RNA sequence in telomerase synthesis of additional DNA copies of the telomeric repeats. Once several nucleotides are added, the RNA template moves down the DNA, and more nucleotides are added. In this way, the telomerase can extend the 3' end of the chromosome without the need of a complementary DNA template. How the complementary C-rich strand is synthesized is not clear. It may be synthesized by conventional replication with the formation of a primer and the subsequent synthesis of DNA. The removal of this primer once again leaves a gap at the end of the chromosome. But this gap does not matter because the end of the chromosome is extended at each replication by telomerase, so the chromosome does not become shorter overall. Telomerase is present and active in only specific cell types that are designed to undergo continuous cell division, such as germ cells, bone marrow cells, and cells lining the intestine. In contrast, most somatic cells lack telomerase, and so their telomeres progressively shorten with each cell division. These cells have a limited number of divisions before their chromosomes become unstable, leading to cell death.
Clear this part? This complication is not clear at all. Why they are adding those repeats there, and why still there is gap? If there is gap, what was the problem? But they are adding the same thing each and every replication cycle, and still there is some gap. Okay. So the question is that telomerase is not present in all cells. Generally, those cells which are going for most divisions, they have this telomerase. Most divisions means that is germ cells, bone marrow cells, and intestinal lining cells. Okay, bone marrow cells, they are always creating that is RBC and WBC and everything each and every time. So they are going for always they are growing. So when we need to study the chromosomes from human or other things, we need to check the bone marrow. Even from the mice, we need to take the bone marrow from human. That is here this bone. And we need to take out the bone marrow, then we can find the dividing cells. That is a very well dividing cells are there, always. But our skin cells, they always don't need that kind of division. When it is required, they will divide. After certain part of the growth of human body, they don't need it. Some cells are dying and some cells are replacing, but the division is not too much. Even just think about your nail. There are cells, they are also growing, but in a very short, very long period. Hairs, they are growing, but taking too long time. It is not required that much. Those kind of cells, they don't have telomerase. They don't have telomerase. We don't know why this kind of division is there. That some cells contains telomerase, some cells do not. But these telomerase, if there is no telomerase at the end, each and every time we have that kind of overlap, a shortening of the chromosome, just like the RNA primer, just the size of the RNA primer. Okay, each and every time, each and every cycle, we have some shortening of the chromosome. Now, finally, if that shortening, so we have the particular telomeric region. Telomere is acting as, if this is a chromosome, and this part is called telomere. Telomere is acting as a gap to protect the other genes of the chromosome. Each and every time, if the cell cycle is going on, this telomeric part is becoming shortened. Okay. Finally, in some case, it will be opened up. There will be no telomere at all. In such case, the original DNAs could be damaged. Original genes could be damaged. and even it can lead to cell death. And this is definitely with aging. So the shortening of these chromosomes into the somatic cells can lead to cancer. And that is the aging and cancer. Okay. Now tell me this one. What do you think? Telomerase is mutated or non-functional. In such case, what will happen? C. So everybody is voting for C. Okay. Now the thing is that, so this is the part, this is the whole replication of the DNA. Now there are two other things. One is that the mutation, that if something would happen to the DNA, if there is some kind of nucleotide change, if there is a change in the certain nucleotides of a gene, then what will happen? We'll discuss this one a little later, after a couple of classes actually. The main thing is that where all the times is happening, that is the homologous chromosomes exchange their segments during meiosis. Exchange their segments means they must have some kind of break and rejoining of the chromosomal segments. That is the DNA. Okay. So based on this one, this is called the recombination break. And the recombination break was first proposed by this scientist. His name is Holiday. Two L's. Okay, not single. This Holiday proposed this thing, that how the chromosomes are breaking apart and how they are rejoining. That's why it is called the Holiday model, Holiday junction model. According to this one, the chromosomes, if that is a single chromatid exchange, that is only two chromatids are involved for two non-homologous chromosomes, then there is a break on each point, each one chromatid, same locus. Okay, always remember that it is in the same locus. Then these chromosomes can join to the opposite chromatids by some kind of mechanism. Some proteins are helping them to join with each other. Now, when these kind of crosses are created there, just that cross is moving towards the end, and in such a way, the other part of the chromosome is coming to the other side. Clear? This way. This cross is there. Suppose the cross is there, it is going like this way. And the chromosomal segments are extended, like just like chiasma. Okay. Now, during when these parts were exchanged between themselves, then they can form this kind of pattern of rejoining. Now, when they can form this kind of pattern, either it can be divided into this plane or it can be divided into this plane. Based on the division, they can form the different kinds of gametes. So main function is that here the breakage in two strands, then the rejoining of the chromosomal segments, that is the DNA, into the other strand, then the Holiday crossing there, and that crossing is moved, and as a result, the recombination molecule is formed. This is one part. Another part is that if there is double strand break, it is a little bit difficult. In case of double strand break, the certain part from the break is removed first. We don't know what, but certain part is removed. Then new DNA synthesis is occurring there into that phase using the other one as template. Okay. So here is the chromosome, and this part is broken here. There is another segment here. Now the synthesis from this part is starting, or replication is starting here using this one as template rather than using this one as template. Okay, so in such case, the complementary strand is forming here rather than this one. And they are exchanging, again the crossing is there, and they are exchanging their parts. Why? What are the molecular formula? Still we don't know. But this is the proposed model for Holiday, that there is a certain kind of replication, and replication is using the opposite strands template other than using his own strands template. And in such case, but there is some kind of mismatch pairing. Just by using the other one as template, there can be some kind of mismatch pairing. Now, suppose A is here and here is C. This is a mismatch pairing. In such case, what will happen? DNA polymerase 1 will come and it will remove that part. After it removes that part, it can take the other one as template or its own one as template. Okay. Based on this replication, they can form either a recombination type or a normal type. So the DNA replication here is just like using this one as template or its own one as template. That is the main thing for these three models. Okay. This is called the Holiday model for recombination. I'll not go too much into details on this. I think this is more or less about the DNA replication. So what are the differences between eukaryotic DNA replication and prokaryotic DNA replication? What are the differences? Main points. Histones in eukaryote. So the replication of histone in eukaryote, then there is no histone in prokaryote. That is one. Second. More replication site in case of prokaryote, there is origin site. More origin site in case of prokaryote, there is generally one or two origin sites. Okay. Next point. You can write it down now. We discuss these points are just in context of replication. Different types of DNA polymerases there in eukaryote, and different types of DNA polymerases there in prokaryotes. Okay, this is the third point. Next. MCM27 complex in case of recognition origin site in case of eukaryotes, and Odyssey DNA complex in prokaryotes for the recognition of origin sites. There is one. Next. Termination in prokaryotic chromosome by TAR complex or TAR sites or termination sites. In case of eukaryotic chromosome, just fusing of two replication parts. Next. Already histone is there. Histone part is done. Next. Telomere end point replication problem. There is no such telomeric part in prokaryotic chromosome or prokaryotic DNA. But in case of eukaryotic DNA, there is a telomere part, and the replication is telomere is different than the original replication, original DNA replication. Telomeric replication requires the enzyme telomerase. In case of normal replication, eukaryotic replication, it requires DNA polymerase. There could be another differences here also. the differences between replication at telomere, difference between replication at original DNA. What are the differences if I consider in this way? Original DNA replication, both are eukaryotic DNA. Original DNA replication versus telomeric DNA replication. What are the differences? Original DNA replication, DNA polymerase is the enzyme, main enzyme, and telomeric DNA replication, telomerase is the enzyme. Original DNA replication, one RNA primer is required, and that is synthesized by primase. In case of telomeric DNA replication, telomerase contains its own RNA primer, so no RNA primer is required there. Generally, no addition of extra part in original DNA replication, there is no scope. In case of telomeric DNA replication, always a repetitive sequence is added. There is no such repetitive sequence in case of original DNA replication. In case of telomeric DNA replication, there are always repetitive sequences. Okay. There is another enzyme, it is not in your syllabus or not in your book. That enzyme is called tankylase. It is also working in the telomeric video. Tankylase. Okay, telomerase and tankylase. One person here in UC, he is the only person who is working with the telomerase. So those who have the project on telomerase, I will suggest in Caroline Price. Her name is Caroline Price. Go to her research work, so you will find a lot of information about telomerase. She is the only person here who is working with telomerase. Once I was very interested about her, but we didn't teach. She is the only person. She is working with tankylase and telomerase both. Okay. So this is all about replication. I will suggest you to go this part very well because this is really very, very interesting and that is important. Next, the part is gene expression. Now go to the gene expression. So DNA replication is complete. Now the gene needs to be expressed. Gene expression means the end production of protein. Finally, the protein synthesis. This model. What is this model called? What is this model called? I told it before. This was proposed by Watson and Crick. This model. What is this model? And you have to remember this.
Structural DNA. Structural DNA. No, the photograph with Franklin.
What is this model called? That is the DNA replication, that is RNA synthesis and protein synthesis as a together. This is called central dogma model. Okay, proposed by Watson and Crick. Okay, so what is this model? DNA is replicated by transcription. This transcribed mRNA will produce a protein, and that process is called translation. Transcription means it is transcribed into same format but in different other way. That's why it is called transcription. And this mRNA is translated into another language, nucleotide language to protein language, amino acid language. That's why this is called translation or protein synthesis. Between these two parts, there are certain other things. After transcription, when the mRNA is formed, after that it goes for further processing. That is called RNA splicing of mRNA. Okay. After this RNA splicing, this matured mRNA will go for protein synthesis. But here the protein is not fully formed. After this protein synthesis, there is another process that is called post-translational modification or protein folding. Okay, then the actual protein is formed. There are several other things. Previously, we didn't know that. Actually, Watson Crick didn't know that there are several other processes, because the question is that we have one gene can translate only one. One gene can translate many proteins. And that is when we'll go for the splicing part, then we'll know about that one. Okay. So what is transcription? The synthesis of mRNA from DNA template. Now RNA is the genetic material, we already know that. And there are certain kinds of RNA. One type of RNA is called catalyzing RNA, and which is called ribozymes. What is the function of ribozymes? They can be self-replicating. They can replicate by themselves. And their function, they can cut their own sequences. They can cut their other parts of the RNAs. Can connect some RNA molecules together so they can replicate others. And catalyze the formation of peptide bonds. So this is the function. They can act as an enzyme. That's why these are called catalytic RNA or ribozymes. But mainly their process is that transcription, replication of RNA in RNA viruses, RNA processing, that is the RNA splicing and other things, and translation. Okay. Do you know how many types of RNA do we have? How many types of RNA? 1,
No,
3, what?
transfer, messenger, and ribosomal.
tRNA, rRNA, and mRNA. Any other? RNA silencing, mcRNA, RNA silencing, do you know that? Okay, the RNA has generally two structures. There is primary structure and secondary structure. These are the bases of RNA. In case of thymine, in RNA we will find the uracil. Okay, you just need to know the names of this one. If you are interested in structure, you can go for that. But in our class, we don't need really the structures too much. So this is the structure of RNA, linear structure. And what is the difference between RNA sugar and DNA sugar? Both are pentose sugar. First type of things, they are pentose sugar, and these are the ribose sugar, whereas DNA is deoxyribose sugar. Okay, addition of this one, actually this OH group, and there is one H group, and as because they have this OH group, they are mostly active. They can act very well and they can be degraded very well as quickly. That's why RNA is not actually staying for a long time in the cell. It is forming and then it is degrading. And that's why when we are working with RNA, we have to use 2 gloves and long coat up to this, because if we are working in this way, from our skin cells, if the one cell can go into the solution, it can release the RNAs enzyme there, and it will degrade, the RNA will be degraded. Totally. So there are 2 structures of RNA. One is the primary structure. This one is the primary structure, that is a linear structure. But in this linear structure, they might have some kind of complementary sequences. When they have those complementary sequences, their always tendency is that to bind with each other. Those types of things, we already discussed that one. They can bind with each other, forming that kind of loop, hairpin structure. Okay. So the difference between that, both are composed of nucleotides, DNA and RNA. DNA sugar is deoxyribose sugar, RNA sugar is ribose sugar, and that OH group in the 2' position present in RNA only, and there is no presence of that OH group in DNA. there is hydrogen instead of that one. Bases AGCT here and AGCU in RNA. Okay. Nucleotides joined by phosphodiester bond in both cases. This is similar. Both, this one is mostly usually double-stranded, but single-stranded DNA is also present in nature. This is usually single-stranded, but in case of certain viruses, there are double-stranded RNAs. Okay. And we're doing HIV. Are you doing HIV? So did you go for the retrovirus sequence there? Not yet. Okay. Okay, their secondary structure for DNA is double helix, but in case of RNA, many types. It could be linear, could be double-stranded, could be double helix, could be hairpin, or lot. When we will go for the tRNA, we will see that there is a lot of things. And stability, generally DNA is stable, very stable. DNA is very stable, and that very stability is from Jurassic Park movie. Okay. That DNA was very stable. Yeah, that is the based on science. That was based on science. And not only that, there are a lot of science there. And the science was that some parts of the DNA was missing for the T-Rex, particularly for the last one, that is Indominus Rex. Okay. When they found that those dinosaurs, what was the short one? I forgot the name. What was the short one? Velociraptors. Velociraptors were communicating with these Indominus Rex. Why? Because some part of the DNA was missing. Those DNA was from Velociraptor DNA. Then they find out. So science is there. Everything is there. Though that is the movie, but a lot of science is there. Okay. Even in the Spider-Man movie, the Spider-Man got the radioactive spider's bite, and that changed his DNA. It was in the first part of the movie. That is changing his DNA structure. So science is there always. And that is true that that DNA was isolated from the mosquito blood. Mosquito took that blood from the dinosaur, and that DNA was isolated from that fossil. We can do that thing. Fossil DNA is still active. That is, you can get every type of composition from there. DNA is very stable, but not RNA. It is easily degraded, and generally we cannot find too many RNA in our body most of the time. When it is required, it is produced. Okay. Now, these are the RNAs. These are the RNAs. So one is ribosomal RNA, rRNA, messenger RNA, mRNA, transfer RNA, tRNA, small nuclear RNA, snRNA, and small nuclear ribonucleoproteins, snRNP, small nucleolar RNA, snORNA, small cytoplasmic RNA, scRNA, microRNA, small interfering RNA, and PUE interacting RNA. This one is right now, siRNA is very, very interesting right now, because using siRNA, you can do the gene therapy. Okay. So these are the main classes. I divided everything into two classes. One is the main class, that is the messenger RNA. It is different functions. That is, tRNA is required for protein synthesis, but it is not actually transcribing the codes. The transcribing the codes is only mRNA, the messenger RNA. Okay. rRNA is responsible for formation of ribosomes and other things. I'll show this one, the next one. So ribosomal RNA, mostly those three things, ribosomal, messenger, and transfer RNA, they are prokaryotic and eukaryotic. In case of all other RNAs, mostly they are eukaryotic. Only this one is prokaryotic, that is the CRISPR RNA. Do you know CRISPR gene? CRISPR is actually present in case of some kind of plants, generally rice and wheat. So using this CRISPR gene, we can modify the genetic modification there and other things. Later we'll talk about this one, CRISPR gene, when we'll do the agricultural genetics, that part. Okay. Now, these are mostly the mRNA is in the cytoplasm and nucleus. These two are nuclear RNA. This is in nucleus and cytoplasm, and all other mostly in nucleus and cytoplasm. Okay. Functions. This carries the genetic code for proteins, mRNA. Ribosomal RNA is the component for ribosome. Ribosome has 2 units, and those 2 units are produced by this ribosomal RNA. Do you know ribosomal RNA has, a ribosome has 2 units? Do you know the structure of ribosome? Doing better, but ribosome has 2 units. One is small subunit, one is large subunit. Okay. These subunits are separated before protein synthesis. During protein synthesis, they are attached with each other. Eukaryotic RNA, a eukaryotic ribosome contains, this one is 30S and this one is 50S. As a whole total is called 70S. That will be in your question. Okay. In case of, this is the prokaryotic, and in case of eukaryote, it is 40S, and this is 60S. As a whole total is 80S. Didn't you read this before in your courses? The cell structure, cell components? No? Okay. Now the math is not clear, right? 30 plus 50 would be 80, but it is called 70. Then 60 plus 40 will be 100, but it is saying 80S. S means Wedberg unit. Wedberg unit or Wedberg sedimentation unit. Wedberg sedimentation unit. So it is not like that. If you can isolate those components and you can spin it, then the sedimentation rate of the small subunit is 30S, and the large subunit is 50S. And as a whole together, when you can spin it, the sedimentation rate is 70S. That's why it is called like that. There is no math. There is a 70S like that way. These components, these subunits are made up of rRNA, ribosomal RNA. The main component is rRNA here. Okay. Transfer RNA, that is the main part for protein synthesis, which is helping to carry out the amino acids and which helps to code, actually read the code and add the amino acids according to that code. And all of the RNA, they have some other functions. We'll not go for those things together. Okay. Now, what is transcription? Transcription means the synthesis of RNA from a DNA template. So we need a DNA template to transcribe the mRNA. That is the first thing. DNA template is required. The code is transferred from DNA to RNA to protein. Okay. Now this template strand is required. Who will transcribe that? There are some enzymes are required. First, one enzyme is required, that is the RNA polymerase, who will transcribe these things. But in DNA, there are some regions there. Whole DNA or whole gene is not transcribed. There are certain portions. Like if the DNA is like this portion is called the promoter region. Promoter region means that is the binding portion of RNA polymerase. It is not transcribed. Okay, that is the promoter region. Then the RNA coding sequence. Here from this part, RNA will be coded. And the termination site. Not only that, there is another one. When we will go for the gene regulation of gene expression, we will find that after this promoter sequence, there is another part which is called operator. That operator controls whether the RNA polymerase will go for the synthesis or not. Okay. So now from this just up to this, that is the DNA is containing this kind of promoter region, RNA coding sequence region, and termination region. Based on this one, if I know the sequence of RNA, can I know the sequence of DNA? if I know the sequence of RNA, can I know the sequence of DNA? Yes. 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 No. No. Why not? Because RNA is produced from this part only. We don't know the sequence of operator, we don't know the sequence of promoter, we don't know the sequence of terminator. Only this part. So if this is a gene, this is the whole gene. So if I know the sequence of mRNA, I cannot know the sequence of the whole gene. That is the problem. And that is bioinformatics here. You can know the sequence like this way. How? If I know the sequence of RNA, I can know the sequence for this RNA replicating part of the gene. How can I know that? Suppose this RNA is can I know the sequence of DNA from here? How? Certain concepts. How can I know the sequence of DNA from there? This is the sequence of RNA we have. I need to know the sequence of DNA from here. This is bioinformatics. You can just use it that way. Later, when we will know about the translation, this AUG part, this is called a codon. You can synthesize one amino acid, that is methionine. If I know that the methionine is there, I will know that in the RNA, AUG is there. Okay. This is omics. Do you know about omics? Genomics. Do you know what is genomics? Proteomics. What is proteomics? Proteomics means the finding the structure of protein there, not only structure of protein, the sequence of protein. Based on the sequence of protein, try to find out the sequence of DNA. This should be complementary base pair. Always complementary base pair. So if this is the base here, and the DNA sequence will be A, there will be T, there is no U there, thymine will be there. If U is there, then it is A. If G is there, then it is C. If C is there, then G, A, and T. That is the sequence of this strand. If this is the sequence of this strand, what is the sequence of the other complementary strand? Think about that. T is there, that is EA. A is there, that is T, C, G, C, T, and A, right? What is the similarity between these two? Is there any similarities there? Except you. Everything is same except you. And that is the concept for bioinformatics. If you know the sequence of mRNA, you can find the sequence of DNA. Such a way. That is the concept of bioinformatics. But the problem is that we will not know that part of this. And there are some other things. It is not so easy. There are some other things that that is transcribed into RNA, but that is not used. This part, mRNA, is called transcriptome. Okay, this is called transcript. This is sometimes called transcriptome. T-O-M-E, transcriptome. That's why reading the sequence of mRNA is called transcriptomics, rather than mRNA genomics or something like that. It is called transcriptomics. Reading of protein sequence is called proteomics. Reading of mRNA sequence is called transcriptomics. And reading of DNA sequence is called genomics. Omics. This is the modern biology right now. Omics. So this is the main thing here. Some RNAs are transcribed in both prokaryote and eukaryote. Those are the messenger RNA, mRNA, rRNA, and pRNA. In case of eukaryote, these are the sets of RNA which is produced in eukaryotes only, but not in prokaryotes. And they have different functions. And this one is only for not prokaryotes, only for prokaryotes or generally plants, but not for other eukaryotes, animals. And we can find that this is a structure electron micrograph of a DNA, which is showing that transcription is going on. So when there is transcription, these mRNAs are produced from here. That means this part contains one gene. Okay? Or this is the whole gene, and each and every time there are several cycles of transcription is going on. It can be that is possible. One gene can produce several mRNAs at same time or different time, because a lot of protein is required during that time. And if the mRNAs are produced in this way, we can find this leaf-like structure. And that is showing us, okay, that is transcription is going on into this gene or DNA. Now the question is that DNA has double strands. Okay, mRNA is single strand. It is producing from the DNA, whether both the strands are working or not. Now think about this one. This sequence, this is the complementary sequence of DNA, the other strand, and this sequence of mRNA is same. This sequence is formed, this mRNA is formed from this part of the DNA, this segment of DNA, this strand of the DNA, not the other strand. Okay? If this strand will produce the mRNA, will synthesize the mRNA, their sequence will be different. Now the question is that, but this is a part of the gene. This whole part is called the gene. So double-stranded DNA is there. So what is the function there? They will produce two different kinds of mRNA, or that will be two different kinds of mRNA, definitely, because their sequences are different. Whether they are producing two different kinds of mRNA or just producing only one mRNA from one gene. Clear the question? Question is that DNA contains double strands. Okay, two strands are there. So my question is that whether mRNA is produced from only one strand or from both strands. If this is called the gene, gene is for only one character. In Mendelian genetics, we have seen that gene means only one character. Different traits are there, either tall or short or whatever it is, but that is only one character. If from this strand the mRNA is produced, it has a different sequence, and if the mRNA is produced from this strand, it will have different sequence. The question is that whether they will produce those two types of mRNA or a single type of mRNA. Any idea? No? They are using only one strand. Only one strand. Okay. Which one? We don't know. And that strand is called template strand. From the strand of DNA from which the mRNA is produced, that is called the template strand. The other strand is called non-template strand. And from non-template strand, there will be no production of mRNA. Okay? Now the question is that, another question. Question is that, this is the DNA. This is the whole DNA structure. There are three genes here. If this is the non-template strand, this is non-template strand, and this is template strand. So in such case, all the mRNAs will produce from this strand, or they will use two different strands. Otherwise, one DNA strand is non-functional, right? Actually. whether they will produce from only one strand or different strands. Clear the question? If only this template strand will be used, then this strand has no function at all. Non-template strand. The answer is that they can switch at random. Suppose these two genes are they are using these two part for two proteins, but this part from one protein. These two part of this template strand. In such case, this is the template strand, this is template strand, and this is non-template, this is non-template, certain part, another part is template, and here it is non-template. But only one will be used. Okay, and this is the structure. So gene A, gene B, and gene C. There are three genes here. Any one, here is the mRNA formation. So particularly for gene A, this is the template strand, this is the non-template strand. For gene B, this is the template strand, but this is non-template. With the same strand of the DNA, there is a template part and non-template part. Okay? And this is the way the mRNA is formed. Now, there are certain parts. One is called the promoter. Promoter, the function of promoter is to attach the RNA polymerase onto this DNA segment, DNA template strand. Okay, that is called the promoter region. Generally, promoter region is a little bit upstream. It is not really should be that just attached to the gene. It could be more upstream also. Okay, there is more upstream part there, then certain part which is non-functional, then actual functional part is there. And from this functional part, that is called the RNA coding region, the mRNA will be produced. And then another is called the termination site or transcription termination site. When the RNA polymerase will come here, it will terminate the synthesis. Now, which of the following phrases does not describe a function of the promoter? B, D. Promoter. Promoter means when we are naming something, I told you several times, when we are naming something, we are thinking about the function of that one. Polymerase, the enzyme which can polymerize. Nuclease, which can cut the nuclear part, that's nucleotides, that's called nuclease. Promoter promotes something. If it promotes something, it will not end that one. It will start something. That is the promoter. Which terminates the thing, that is called the terminator. Not the SWOT generator. That is the terminator. Okay. So these are the things. So what is these things? Serves as the sequences to which the transcription apparatus binds. Binding of RNA polymerase onto that site. That is called the promoter. Oh, it is here? No. That is wrong. I have to change that one. Okay, so just I'm giving the initiation of transcription, the first part. There are three phases, just like the DNA replication, three phases. One is initiation, then elongation, and then termination. Initiation means first stage of transcription. The function is that binding the correct RNA polymerase onto the promoter site. That is the first part. In case of DNA, there is origin of replication, binding of originator protein onto the origin of replication, and helicase protein, that is the initiation, single strand binding protein, all those things was the initiation site. Here, mainly the two things, correctly bind the RNA polymerase onto the promoter site. That is the initiation. Okay. Elongation means elongation of the RNA chain by adding RNA nucleotides, not the DNA nucleotides. So ribonucleotides should be added, that is the elongation. Then termination, the last stage of transcription, needs to release the RNA chain as well as RNA polymerase enzyme. It is little different than DNA replication. Okay. There are certain proteins there as a whole, the whole machinery will be coming out. So yeah, we are done for the day. What is replisome?
What?
Replisome. What is replisome? You're laughing. What is replisome? Because when I saw this one, the whole machinery, then I thought that okay, we had some kind of this term before. The whole machinery. It's called replisome. What is that? These will be the questions, okay? The total, yeah, what are the subunits of replisome? What is replisome? Though it is multiple choice question, maybe, but you have to know about this. What is replisome and replicon? What is replisome? Total, the whole machinery for DNA replication. Whole machinery means what are the things there? Helicase, zydase, single strand binding protein, primase, forming the short RNA primer. These are the main things. And as a whole, this machinery is going up and the DNA is synthesized. Okay. Then a lot of that. Okay. I think everything is okay.